Yes. So, welcome to our only one analytical technique talk series. And today's session is the ninth talk, which is the focus is on biophysical characterization, which will give us insights into the biomolecular, intricacies of biomolecules. This session is hosted by the analytical services team at Venture Center. For those who are attending our session for the first time, I'd like to introduce you all to Venture Center. Venture Center is India's leading technology business incubator, and we support deep tech, science-based startups. We are a Section 8 not-for-profit organization, and we are hosted by NSTDB and BIRAC, a Government of India entities. So, since we support science and deep tech startups, our objective is to provide a complete ecosystem. So, what does this ecosystem comprise of? So, we have various components of these ecosystems. So, some other, we provide mentoring of service. Then funding, we connect to various funding agencies. We have the infrastructural facilities, which comprise of dedicated labs, offices, and then we have various funding databases, and then we have partnerships with different agencies who provide the design and prototyping facilities. Then we have the manufacturing facilities, finance, legal, HR, so on and so forth. Coming to the heart of the technology business incubator at Venture Center are the scientific and the analytical facilities. So, as part of this, ecosystem, we have certain scientific facilities which are called as the plug and play labs. So, we have 100 plus very workhorse kind of equipments, and they are available. They are open access, and anybody of you who require these can come and use it. Then we have a well-established prototyping facility for all those who want to build their prototypes and carry out certain additional testings that they can be done over there. And then we have the testing and analysis horizontal, which comprises of some core analytical equipment and some specialized and high-end equipments that are housed here, details of which I'll be explaining in the upcoming slides. We have a regulatory facilitation center, which provides mentoring and hand-holding to various startups and other industries. And we also have a tech transfer and IP group who help file patents to the patent landscaping and other related activities. And we are implementation partners for various national programs. Coming to the analytical services at Venture, I mentioned, we have some very core analytical equipments like GCs, HPLCs, FTIR, elemental analysis, particle size analysis, and all of this is available for not only for the startups, but for anybody who may require of carrying out, maybe you require it in your research or in your quality control. So these facilities are available to all. Then we have some high resolution mass spectrometers. We have QTOF systems and GC headspace system available at Venture Center. All of these are open access, and as I mentioned, all the details are available on our website. Everything is available there, and you can reach out if any of you all have any specific requirement. These are open access facilities, and we have special discounts for startups and for students. Coming to our latest, addition to our analytical facilities is the Center for Biopharma Analysis, which is a GLP compliant and NABL accredited facility. This is supported by the National Biopharma Mission and the BIRAC, both Government of India entities. CBA, as we call it, is a one-stop destination for the characterization of biopharmaceuticals. We are one of the few facilities in the country where we provide both the structural analysis and the functional analysis under one roof. We host all high-end dedicated instruments, and we provide hand-holding and advisory support to biopharma researchers and anybody else who requires these services. All our services are listed on the website, and I'll also be sharing the details in the chat box. So this is a list of the services that are offered at CBA. So on the structural side, right from the primary structure to the higher order structure and biosimilarity assessment, we have a whole suite of services, and the team has done almost 15 plus molecules we've characterized in the recent few years. And on the functional assays also, we have developed very many assays, and now we are in the validation phases. So details are all on the website, and any of you all who require this can reach out to us. I'll be sharing our contact details in the last slide. So as CBA is a GLP compliant facility, We have all our equipments that are qualified and are 21 CFR compliant. So this is a list of the equipments that are available there. So we have one more HRMS system, then we have FTIR, SPIR, SPR, CIF system, fluorescence spectrophotometer, SEC, RI MAL system. Then we have the standalone HPLC systems, nano DSC, CCD, and stability chamber. So with all of this, we structural characterization. So with that brief introduction about the venture center and our analytics, I'll now move, to the speaker for today. So we are very happy to have with us Dr. Vadiraja Bhat, who has been gracious enough to accept our invitation. He is the country Biopharma Business Development Manager at Agilent Technologies India Private Limited. Dr. Bhat comes with a PhD in chemical biology from JNCSR and a postdoc in proteomics from MIT, USA. He has several years of experience in biopharma research and development. He has held significant roles, including the senior LCMS application scientist at Agilent's USA facility. And he's also been the associated director at the proteomics lab at Baylor Scott and White Memorial Hospital, again in USA. He has contributed to advancing LCMS workflows for proteomics and many biopharma applications, and has been an adjunct faculty member at the Manipal Center for Biotherapeutics Research and the Manipal Academy of Higher Education since 2023. So with that introduction, I welcome Dr. Bhat to now deliver his session. Sir, over to you.
Thank you. Thank you, Edna, for the invitation and the kind introduction. You can hear me, right?
Yes, you're audible.
Okay.
Request if you can please share your screen.
Yes, I will share my screen. And when you see my screen, let me know.
Yes.
Okay, I'll put into, yeah. This. Yeah, once again, thank you. So today I'm going to talk on biophysical characterization. That's the tools for revealing biomolecular insights. Essentially, my talk is mainly focusing on the like some of the analytical tools that will be used for characterization of the biomolecules. Main focus going to be on uh, uh, main focus is going to be on monoclonal antibodies, maybe like a little bit touch on the other molecules as well. So when you look into the biomolecules compared to the small molecules, of course, in between we have the complex drugs as well. So of course, small molecules, everybody knows, they are like really small and simple, and molecular weight, start like maybe 100 to 200 daltons, so it can go to 1500 daltons. But like when you look into the complex uh, can say complex small molecules or complex drugs. They are like oligonucleotides, maybe synthetic and recombinant peptides, and up to the insulin level. They are not that complex compared to the large molecule. So then you have like large biologics, small biologics, and large biologics. Small biologics are like an essential, the smaller proteins, and large biologics generally what we call is monoclonal antibodies, ADC, fusion proteins, and of course, bigger ones are cell and immunotherapy. Now, the small molecules compared to analogy is like really simple things like bicycle. It's just analogy, right? So you can look into that if you want to see analogy or if you want to look into say characterize a bicycle, may not be that difficult, right, for anybody. I can dismantle and put it together, and anybody can do it. But when you look into the car, just you can say complex or complex drugs, or say smaller peptides and smaller proteins, like a car. Still can manage, they're still complex but not that complex compared to the aeroplanes. Now aeroplanes are like your monoclonal antibodies and bigger proteins, and you can go to even complex cell therapy, it's like really huge like jumbo jets, very complex, right? So that so many things are involved for the safety and efficacy. Now, when you look into the market side, uh, biologics, of course, gaining the market share, of course, they are growing compared to small molecules, much more rapidly compared to small molecules. when you look into the different therapy areas, like here it's like showing the top 10 therapy areas. So obviously, oncology is the number one by looking at that size, like size of the bubble. But if you look into the say the highest growth or CAGR, you can see that for next few years is gastrointestinal. That is essentially like what we're talking is uh, uh, the peptide GLP-1 related peptides. Because of that, the highest growth rate, even though oncology is still going to be remain the largest category. Now, as I mentioned, the biologics going to catch up with the small molecules. In next few years, probably they're going to almost like 50%, right? They're almost like, uh, 58, 42%, but it's going to be, close in the coming years. Now, look into the peptides. I just want to give just a very brief into peptides. So, because it's a really high growth market, now if you look into the particular GLP-1, look into that. So it's probably GLP-1 is nothing but endogenous peptide. It's like a it's already there in everybody's body, but of course, and therapeutic side, exenatide actually essentially is extracted from the initially extracted from the lizard. It's called Gila monster. So essentially, it's called extendin 4. So essentially, it's a small quantity, and so that obviously it's not enough for therapeutic. So that it has to be synthesized chemically or recombinant to named it as exenatide, introduced in 2005. Following that, a lot of researchers are done in this particular molecule, a lot of changes, as you can see in the right side. And now we have uh, look into that. The half life is 2.4 hours to once a week one can take. So, so essentially, also world formulas are available. So essentially, this is going to be a huge market. I don't show here the market, but it's probably this year and next year it will be globally around GLP-1 related market size, probably around 50 to 60 billion dollars in this year and next year. So it's a huge market. And when you look into the protein therapeutics, because today I'm going to focus on that. So let's look into the monoclonal antibodies generally speaking, and there are simple fragments of the monoclonal antibodies to pegylated uh, Fab and fusion proteins, bispecific, multispecific antibody conjugates, and also other fusion proteins. So there are many things are available, and more and more complicated or complex maps or monoclonal antibodies are added into the into therapeutic area like bispecific and multispecific, along with the bispecific multispecific drug conjugates. So it's even making it more complicated. As I mentioned earlier, cancer is a major therapeutic area, and if you look into that cancer alone, there are so many types of biologics available today, right? So you can look into monoclonal antibodies. Of course, cell therapy is a growing area related to cancer and maybe other areas as well. And of course, the ADCs, particularly related to cancer, and also radionuclides. So that there are many things are available for the just cancer alone, and that's why it makes the one of the largest category within the biopharmaceuticals. Now, uh, the within the ADC antibody drug conjugates, if you look into that, oligoconjugation is gaining more attention because oligotherapeutics are growing, and oligotherapeutics generally, if you give without any kinds of particularly targeting the organs or tissues, it will directly go to the liver and getting degraded, metabolized, and it's gone, right? So if you want to target to the particular organs or tissues, then it has to be directed. So the monoclonal antibodies are really good directed to the particular organs or tissues or cell types, so that it can enter into the particular cells or organs or tissues, and then it can act as supposed to be. So that's why the antibody oligoconjugation or peptide oligoconjugation is called AOC or POC. So they are actually really gaining attention, and it's going to grow in next few years. Now, when you look into the antibody conjugate, and also people are considering the multispecific because they're looking into multiple antigen targets, and so that or bispecific, multispecific, along with the small molecules. So when you look into the analytical side, it's going to be really complex. And now people are also looking into the multi-payload. That means different types of drugs in this antibody. That means if you consider bispecific and multi-payload, that molecule is like a really complex for analysis for any analytical scientist, right? So it's a growing area, and definitely in coming years you'll be seeing that more and more people talking about these kind of very complicated molecules along with the bispecific and multispecific maps. Now, monoclonal antibody itself is very complicated. So let's look into the just very simple, very briefly. So monoclonal antibodies is like everybody knows that you study the biochemistry or molecular biology or cell biology in everywhere, like you might have studied the structure of monoclonal antibody or antibody in general. It's like a Y-shaped and it has light and heavy chain, and it's a molecular weight of around say 150 kilodalton, right, roughly. Now it has different parts and generally called a antigen binding site. It is the Y part of the antibody that's called Fab region, and also effector region called FC region. Now, like within the monoclonal antibodies, there are many things are there, right? One is like generally glycosylation. So glycosylation is very important for its function, and also like if you look at that to hold these like heavy chains together, there is a disulfide bond. Also, there is a possibility of the shuffling of the disulfide bonds can happen, and so that can have impact on the structure and function. Now, it also has the modification, like it can be pyroglutamate at the N-terminal, it can create heterogeneity, lysine, in the C-terminus, it's a, addition, one or two additional lysines or truncation that have that is also like, going to get different, um, uh, forms of the monoclonal antibody. And of course, like, it can fragments and aggregates, right? So all of them have the impact on the, immunogenicity and safety. So have a particularly efficacy as a potency as well. Now, if you look into that, it has a the structural heterogeneity potentially have impact on many things, like including the binding to, as I mentioned, immunogenicity or effector efficacy and its kinetics, as well as pharmacokinetics, and of course the mechanism of action. So we are not going to talk all these things today, but we will focus on to the like essentially some of the questions one can ask on the monoclonal antibodies. So let me get a pointer here. Okay, so monoclonal antibody. When you look into the monoclonal antibody itself, there are many questions one can ask. Like if, any analytical chemist or scientist or those who are working on this monoclonal antibody characterization, they have many questions, right? So intact mass, like are you working in bispecific, multispecific maps? Like what is the glycoforms, quantitation, vector sequencing, primary sequence, and particularly post-translation modifications including the glycans. Disulfide bonds, whether it's scrambled. If you're working on the ADCs, like or oligonucleotides, what is the DAR? Antibody drug conjugate ratios, antibody drug ratios, or drug antibody ratios. Like glycan, particularly very important, and glycan, what is the glycan profile? Intact, subunits, as well as peptide level, and also like release glycans. So if time permits, we look into some of these things. And of course, like nowadays people are talking into more into native analysis of the proteins, particularly in mass spectrometry. We will look into that, some of these things in coming slides. So, and also non-covalent protein complexes, also one people are looking into that. There are many questions, not only these, but there are many other questions. Now, if you look into the regulatory side, ICH guidelines are the number one, right? So that everybody talks about ICH guidelines. There are like a quality, safety, and efficacy and multidisciplinary guidelines. There are about 70 guidelines and available, and it's continuously revised, and newer guidelines are revised guidelines, and also sometimes draft guidelines keep on coming. And if you look into that, within that 70 plus or more or less 70 guidelines in ICH, so like a quality 24, safety 15, efficacy 21, and of multidisciplinary 9 guidelines. When you look into the particularly the guideline side, so there are many different categories. Right? Everything starts with the ICH guidelines with the Q1A like that, and it can go to like Q12 or more. So essentially, like all of us know that Q6A and P is very commonly used for the analytical side. Now, when you look into the analysis of this monoclonal antibodies or protein therapeutics or any molecule, it's called a critical quality attribute. Okay, and when you look into the critical quality attribute, there is a criticality, right? So why we have to analyze them, and why we have to analyze so many of them of the attributes, right? So, like when you look into the product, that's monoclonal antibody alone, there are many things that, like as I mentioned, glycosylation variants, glycan variants, lysine variants. So many things are there, right? And so essentially, like and of course, aggregation and truncations, they are impurities, essentially product-related impurities, product-related substances, impurities, and of course, like we are not talking today, but also process impurities. So when you look at the criticality, aggregation and truncations or the glycosylation, they are very critical compared to the other PTMs, and maybe like even deamitation or even lysine variants are not that much of critical to the safety and efficacy of the monoclonal antibodies or protein. Let's look into the some of this biophysical, mainly into biophysical characterization or analytical characterization of monoclonal antibodies. Okay. Primary structure, number one, right? So everybody want to know that what is the primary structure of the monoclonal antibody, including the glycans and glycan variations, right? So hierarchical structure, secondary, tertiary, and quaternary structures. There is a different types of analytical instruments are going to be used. I'm not going to touch on the secondary or hierarchical structures. Maybe a little bit I'll touch, but not too much on that. So biological functions, of course, is important, but not going to talk today. And of course, like it's kinetics, functional assays, we're not going to talk stability, not going to touch on that. And also, of course, like, the process related impurities definitely are not going to talk today. There are all different types of topics and it can be discussed in different days. When you look into the biophysical properties of monoclonal antibodies, so as is shown in this in the circle, one want to know that what is the purity, what is the, either in fragmentations, aggregations, the thermal unfolding, essentially very important, and of course, like to the stability and also serum stability, glycosylation, and also like charge heterogeneity, and of course solubility depends on the concentration as well as aggregation of the monoclonal antibodies. Let's look into the like some of the important biophysical characterization. As I mentioned, stability, structure function relationships, quality control. Uh, they're important. Why it's important, right? And then if you look into the key physical properties assessed, yes, as I mentioned, higher order structures, aggregation, thermal stability, hydrophobicity, net charges, charge variance, and then the interaction with other molecules. Now, techniques generally used, spectroscopic methods like CD, FTIR, UV spectrophotometers, fluorescence spectrophotometers. Some of the spectroscopic methods are used. Chromatographic methods very commonly used, the size exclusion, ion exchange, and reverse phase HPLC methods or chromatographic methods. Light scattering or scattering methods like DLS, uh, uh, analytic autocentrifuge, checkmarks. So these are used for the higher order structure or aggregations or fragments of these molecules. Colorimetric method, colorimetric method, or like a DSC. Of course, the mass spec we'll talk more on today, particularly because of the it's more and more getting important because it is called multi-attribute methods, and essentially the one instrument can do many things simultaneously. Now, assessment of the biophysical properties, as I mentioned, higher order structures, essentially like different types of instruments can be used, like a CD, FTIR, NMR, HDXMS, all of them can be used for the different types like primary, secondary, and tertiary quaternary structures. So now thermal stability is essentially like the unfolding of the and loss of its native structure. It can be used by the DSC, essentially like looking into the melting temperature. So the interactions, people are like I mentioned in venture center, like they have like instruments like SPR, is looking into the essentially ligand protein interaction or therapeutic ligand interaction. Essentially, like we are looking into that. Aggregation very important, and different types of instruments are used, including the chromatographic size exclusion chromatography, or like the light scattering or multi-angle light scattering, TLS, dynamic light scattering, or analytical autocentrifuge. Of course, the glycan heterogeneity is very important and measured at different types, different levels, intact subunits, peptide level, and release glycans using many of the types of analytical instruments, including the mass spec, capillary electrophoresis mass spec, and of course, a capillary electrophoresis like a CIEF, lectin-based microarrays, and of course, like the typically used helic with separation fluorescent detectors and with very complex glycans. We're also using like multi-top and other types of instruments. Now, I'm not going to go through the details of this. This is already discussed. The molecular characterization, functional assessment, and also effector functions. So that there are different types of, functional and analytical or characterization analysis will be done for the any of the biomolecules, including the monoclonal antibody, and of course other molecules as well. So when you look into the monoclonal antibody, as I discussed earlier, so that when one will be looking into many things, right? And HPLC, essentially I changed here is a critical quality attribute. I mentioned here product quality attribute because it's a product related in attributes what we are measuring. Critical quality attribute, it can be both product as well as process related things. Now, in a monoclonal antibodies, what you're looking is like many things, like charge variants, essentially like when you're looking, we look into the details, aggregates, like how aggregation forms and how it will be measured, and of course primary sequence and peptide mapping, and glycan analysis, oxidation, and of course like your PTMs and everything else, right? So all of them are used traditionally by using HPLC based methods, right? That including, still like people are using charge variants and aggregates, and all these things are done by the typically different types of chromatographic columns and either UV based methods or fluorescence based detectors are used in the backend. Now all of them can be used or analyzed in the LC mass spectrometer. Like simplest mass spectrometer, like single chord mass spec, there are low resolution mass spec to really high resolution mass specs in between, mid range, like time of flight mass specs also come into picture. Simple time of flight instead of the hybrid instruments. Now, this is like one of the reference from 2013. So essentially, like it's showing that what are the analytical instruments we used for the different types of critical quality attribute analysis of the monoclonal antibodies or any protein therapeutics or any biomolecules in general, right? So primary to second or higher order structures as well as like when you go to the PK or you have the quantification or quantitation. So when you look at the mass spectrometry, it comes into picture everywhere, right? From the intact to all the analysis of the monoclonal antibody or proteins or biomolecules to, like peptides and, like glycans and other things. And also somebody want to analyze the small molecules, like, including the polysorbate and the cell culture media, leachables, and as well as, like you want to host cell proteins or, host cell proteins are like in peptides, but like even glycan profiles, once you release them, they're like a small molecule. And of course, fragile as well. Now, traditionally, look into that, many of these critical quality attributes are done by various types of analytical HPLCs or UHPLCs. And one HPLC is dedicated for one type of analysis, typically. And you'll be having multiple number of instruments in your lab, and it will occupy the space, and also more number of people have to be dedicated for this analysis. Now, when you have a mass spectrometry and it can be combined, many of these tests in the one instrument, and essentially like if you look into the like say how we can like make it like a more economical is like reducing the HPLC and adding more mass spectrometry. As I mentioned, simple mass spectrometry like single chord mass specs can do many of this work, so that you don't need the high resolution mass spectrometry for this type of analysis in a routine analysis in a given day, unless you want to do a deep characterization on a daily basis. Now, what is multi-attribute methods? Essentially, like you like develop a mass spectrometry based characterization methods in the R&D lab. This is essentially R&D work. And then once you have that, and you can create a database, and then you can actually establish a monitoring methods. That's not characterization, but essentially monitoring methods using a simple LC mass spectrometry techniques, and that can be done the quality control labs as well. So essentially, the idea is like multi-attribute methods in the R&D lab. Monitoring can be done in the quality control or even in the manufacturing setup as a PAT tool. So not going to cover the PAT tools, but PAT tools also possible for the mass spectrometry. Now, when you look into the the mass spectrometry based characterization of the monoclonal antibodies or any proteins or peptides or other molecules. So essentially, like you need any sample preparation, either manual or automation, followed that you need to separate either, as I mentioned, like chromatographic HPLC based methods or capillary electrophoresis based methods. Either you detect based on your simple UV detectors or fluorescent detectors or light scattering detectors, or you take into mass spectrometry, like as I mentioned, simple single cord mass specs to even high-end mass specs. So essentially, the sample preparation becomes a critical, and essentially they combine all these things, including the softwares for the data analysis and reporting. If you have any of these things, like the step-by-step protocols, SOPs, combine all these can be called as a workflow solutions. So because for example, like you want to analyze a glycan, a release glycan automation or a kits-based approach, separation, then analysis, and then reporting, right? So it's combined as a workflows like this. What you can see here on the right side, I show in the circle green squares. Various types of workflows generally we talk, right? So a typical mass spectrometry lab looks like this. Your mass spec and separation techniques, and of course sample preparation, any kinds of automation you have, or any kinds of automated methods or typical sample preparation methods. Mass spectrometry can be used for the many things: intact, subunits, native, then like vector mapping, PTMs, postal proteins, and of course glycan analysis, right? And glycoforms in the intact and subunits. Now, mass spectrometry also comes with some of the different types of fragmentation capabilities. Typically, like you call CID or high energy fragmentation. And nowadays, all the mass specs has that. Now, like people also like considering like the different types of fragmentations like ETD, ECD, or UAD type of capabilities. It can give different types of fragmentation and a different information, and also like a better fragmentation some of the fragile molecules or like a glycopeptides, or even if you want to do a top-down, uh, these subunits are the entire monoclonal antibody sequencing without any sample preparation. Okay. Now, if you look at the HPLC based methods, it's simple, like as you can see, you're using a cytosystem chromatography and either the ion exchange methods for the charge attributes, size heterogeneity, and also hydrophobic heterogeneity variations, right? Typical people using hydrophobic interaction chromatography. So essentially, it is as I mentioned, one HPLC dedicated for one type of analysis. Typically, if you have a lot of samples to run, and then it's the time consuming limitations, and a lot of work. And now, to reduce all these burdens, one can actually go for like this just showing this schematically how it you are doing that. And of course, if you can add as a mass spectrometry, still you have to go through the same kind of separation. Then generally, like say you are actually, let me go back here, you are doing all this analysis by UV detector, or say some other detector fluorescence, or some other detectors, non-mass spectrometer based detectors. Now one want to get the mass information ultimately for these samples, right? And you want to know that what is the mass of these peaks. HPLC going to your peaks, and the mass spectrometer gives the molecular level information, that is the mass and charge. Essentially, it gives much more information than the HPLC based methods. Now, if you see a charge variant, like you're using a mass spec unfriendly buffers, same thing with your uh, size distribution, or even hydrophobic interaction. So generally, one up like collecting fractions. So like time consuming, and different setups are required for that. Manually, buffer exchange has to be done, desorting. Then if you want to do reduction, and if you want to do further crystallization, something like that, you want to do further calculation, right? A lot of manual work is involved. Now, the drawbacks, as I mentioned, time consuming, like fraction correction, sample degradation can happen, contaminations can happen, loss of sensitivity, data correlation can be an issue, because like manually you have to do certain things. Now you can actually like get rid of all these things, and you can do a online two-dimensional chromatography. That means your second manual work can be done integrated with the second dimension HPLC. So it can be integrated with the first dimension. So we look into some of these things. So briefly talking about that, you are connecting the one HPLC, that's first dimension, with the second dimension HPLC, and doing two different types of chemistry, and then ultimately you are sending the samples to your mass spec detector. It can be two detectors or it can be three detectors. First dimension a detector, that's typically UV detector, and you are collecting particular fractions in online, sending into the second dimension, further separation or desorting, or sending to the mass spectrometry for the analysis. And integration is very simple because the software can handle that. So this is just showing that again, like saying that the mass spec can be connected, simple mass spec like a single quantum mass spec to your high resolution mass specs. Now, if you directly connect without the sample desorting, like let's assume that you are doing the ion exchange chromatography for a charge variant, and it has a salt, organic salt, and what will happen, like nowadays people are using the mass spec friendly salt buffers, so that volatile buffers uh, can be used, but of course, like traditional methods are still with the sodium or potassium salts will be there. So when you don't separate and you end up sending into the mass spec, your mass spec end up like contaminated, and ultimately you will see like what you can see in the red trace. If you don't desalt these your samples, then you will see the data like a red trace here, the MS chromogram, right? So if you desort, you'll see the blue trace, the spectrum. So essentially, like what you have to do is manually desalt your samples to get like a blue trace. It's a lot of work, and manually data correlation has to be done, right? So a lot of work. Now we have done some work. It's done like it's not something new. We have done like in a few years back to look into that. You spike the different levels of sodium chloride, like 150 millimolar, 300 millimolar, and see that obviously what happens, right? So that if you don't use the uh, desalting, then you see like a red trace. If you desalt either manually or online is a 2DLC. You are seeing there's no difference. You added salt, no salt, added salt, no difference, right? So what you are trying to say here is a automation have a huge benefit. Someone want to do the analysis of this kind of the charge-based or size-based separation and analysis by using the mass spectrometry. Now let's look into the particularly aggregate analysis. Okay. Now when you look into the aggregate analysis, like here, they're important, and as you can see here, the monomers are typically one is expecting, but for varieties of reasons, including the temperature related things, or many other freeze and thaw and many other uh, um, uh, issues, it can have a different types of aggregates, right? So mentioned here, like even their sizes, like 40 nanometer, 100, and micrometers, and 10 micrometers, like huge complex, right? So obviously, like as I mentioned, it has a impact, immunogenicity, and uh, uh, essentially, it will be not good if you have aggregates in your samples, and one has to analyze and report, what is the status of the aggregates of these biomolecules, including nowadays for the therapeutic peptides. Okay, and it can be done in many different types of instruments, and it's just showing that how different stages of aggregates formation happens. It's just showing that schematic representation, and also like in different stages when the aggregation happens. Okay, and the N stands for the if no aggregation here. This is what you are expecting, but this can happen like over the time for the variety of reasons. Now, how you are going to analyze them, right? Based on the size of the aggregates, right? So there are different technologies are available, or analytical techniques are available, so that so like as I mentioned, like monomers, any analytical systems will be using, based on the size, you can look into that like simple STS page or a native page, your cycle system photography, all those things can be used. And then the aggregation size going to be increased. Then of course, like you're going to add the like AUC or DLS light scattering kind of stuff. And of course, like when you have like a really visible particles, then you're going to like microscopy or other technologies that's visible because it's a visible particles, right? Um, so now, like just like showing here in this slide, as I mentioned, like different technologies will be used. So of course, like people are using the like typically like the size resolution chromatography or the light scattering, and also of course I will show some information on the how it can be done. I'm not talking about the characterization here, like using the some of these spectroscopy based methods. Maybe I have a little bit of information on the fluorescence based spectroscopy. Let's look into that. Now, traditionally used aggregation by HPLC, UV detector, or combination of UV, RI, that's the refractive index, or the light criteria. So typically, I'm going to skip this slide and the how it will be used technology-wise. Let me look into some of this like the data. So light scattering is a very critical and actually like people are using traditionally for any aggregation data discussions comes. People are using it. It's called SECMOS, right? That's the very traditional use, and it can give a absolute molecular weight, and also it can give size of the molecule. Okay, based on the scattering of the light that you are using laser as a source. So this is an example, one of the recent application notes published by us. So essentially, like here using a 20 angle light scattering detector or multi angle light scattering detector. So along with the side distribution photography, this is just showing that how a monoclonal antibody is called MapTera. It's a rituximab. How it looks like UV that's in the uh, in this peak, you can see that blue trace is UV based, and the green trace is the light scattering. If I zoom that, this is what you can see. The green trace, you can see a lot more information compared to the blue trace uh, uh, in this particular combined detector, like UV multi-angle detector, right? So essentially, what you are missing is really high molecular weight aggregates. Will be missing in the UV, and so that if you want to get a complete information, then you have to go for combination of detectors, because as you can see, the like in the light scattering, still like you'll be missing something. It maybe looks like a small hump, but you'll be missing some of these like fragments, maybe like smaller and not clear, and also like in dimers, right? So that's the light scattering part, and also there's a information I can sharing here is I'll be sharing here is how it can be used in the fluorescent spectrophotometer, right? So in this case, you have to use the label. So once you label your protein, then you can, it's like really like you have a lot of samples to why fluorescence compared to the light scattering or cyclostation based approach or HPLC based approach. The fluorescence-based approach is a complementary to size resolution, and also like in our application we compared how the data looks like to the size resolution versus fluorescence. But the ultimate goal here is one is you have to label in your 9612 plate format, and then of course like you can really quickly scan, and the time is really saved. So that essentially like you'll be saving 75% or more time if you use a fluorescence based a fluorescence spectrophotometer for aggregate analysis. Now this is just a, as I mentioned, comparing between the your light scattering or cyclostation method versus fluorescence method. So essentially, like it's very well correlating with tried for the multiple types of maps, including the NIST map, clusters map, reduce map here. Or if you are interested, look into this application note how it is done and what kind of like this is the fluorescent tag is used, BOPS. That's a big name. So you can use into that, and we can see how it can be done if you have a lot of samples to aggregate analysis. So now this is giving an example of the native aspect, right? So it can be nowadays, as I mentioned, people are looking to native aspect. It's getting very popularly globally. People are like publishing papers and using that for many types of analysis, and combination of columns also. Column chemistry can be used. So in this case, essentially, like I'm showing here, is how a native mass spectrometry used. Why native mass spectrometry? Because you can actually detect the aggregates. Aggregates can be two types, and we'll see that a little bit later. Two types. One is like you can have the covalent, other one is non-covalent. If it is a covalent, uh, if you're denaturing condition, no problem. If you are non-covalent, then of course, the native aspect is going to be the information. So in this slide, you're seeing that your monomer to like fragments to all the way to the dimers, right? So this is the advantage of the native mass spectrometry. A size resolution mass spec friendly buffer can be connected to the a HRMS, and you can get information. Now, a 2D LC can be used, and where you can use a first dimension as a clean up the sample, in this case like cell culture broth, and you can use a proteinase column to first dimension capture the monoclonal antibody, send to the second dimension based on the type of analysis you want to do that. In this application, note and publication, it's like used different types of columns and different types of analysis done in one system. And I'm just showing that, just like again, expanding this native aspect or a denaturing condition, there's a reverse space column used for desorting in this particular case, because the buffers are used, these salt buffers in this case. So essentially, with this, actually you can actually conclude based on the your UV data in the first dimension, and you can say that what kind of pixel you are getting, and in the mass spec you can actually see whether it is correlating with the high molecular weight or low molecular weight. If it is high molecular weight monomer, then it's actually a aggregate. If it is a really covalent, then it's get a really high molecular weight. Otherwise, you get a fragments and other types of analysis, other types of molecular weight you can identify. Now, again, showing that there's another publication of, actually this is a publication published in 2022, so that you can see that the more information you can get, like monomer to non-covalent dimer to covalent dimer. So there is a lot of work is done, including the in India, Professor Anurag Rathod also working on this area, where how aggregate analysis can be done by native mass spectrometry. Now in IIT Delhi. So let's look into some of the charge variants, and with the interest of time, I may go a little bit faster here, but these slides will be available for you. So charge variants are important, and of course, charge is based on the your basic and acidic amino acids, and of course, any PTMs and glycosylation and other modifications can have a you can have the variations in the charge, right? So essentially, there are many things in this graphically in the picture shown that there are many things can contribute into the charge heterogeneity of the monoclonal antibody or any protein in general. Now, it is required according to the guidelines Q6B, so that one has to analyze and submit that this information to the regulators in your dossier. Now, it's showing that the uh, like when you run on the size restriction or the sorry, on the ion exchange chromatography or column, so based on the its nature, if it is basic, then it will be in the on your right side, and just stick on the left side. If it's more so like that, you can see that. Now, okay. So the other techniques generally used is capillary electrophoresis, typically for the charge variants. That's very commonly used, particularly with if it is a really heterogeneous, then you need a the capillary electrophoresis. So just comparing here some of the other methods like capillary zone electrophoresis. That is essentially you can see this is from a publication published on 2015. So essentially, like, if you look into a same protein in the different ways, the CZ, that's a capillary zone electrophoresis, how it looks like. The main peak shows at 28%, 28.6%, and where you don't use the no ampholytes. Uh, then if you use the traditional ionization chromatography, we can actually see a main peak is around 64%, but it's not very clear. But if you use a CIEF, ICF, there's an image, there's a U detector that's called imaged CIEF. So essentially, like with the ampholytes, so actually you can see very close. The main peak is very close to your ion exchange chromatography. So, and of course you can get the both acidic and basic variants here. Okay, so this is a traditional charge variant analysis we are seeing. This is done by the 2DLC, so that you're actually you can directly without any manual desorting, you can actually like really detect these various peaks can be identified in the mass spectrometry, and you can see that what is the mass peak. You can see that there is a differences, but mass it gives the even further information on the glycan variants. Not only the charge variant, but also gives you a glycan variants, right? So it gives you two different types of heterogeneity at one shot. Now, there is a nice publication from IIT Delhi, published in 2022, combining a hydrophobic interaction along with the ion exchange. So that they have done two types of separation. First dimension hydrophobic interaction, second dimension is a ion exchange chromatography, followed by a native mass spectrometry in this case. This is a not reduced, but it's a non-denaturing condition. It's a native mass spectrometry. So they have compared with the without 2D and with the 2D, and obviously they are showing that, say, if you use a hydrophobic interaction mass spec or ion exchange weak cation exchange mass spec, you can see. But if you do a combination of HIC ion exchange, you can get actually a more variance. It's because of many things. One is like a improve the sensitivity, second one is stability, so that the low abundant variants won't disappear, so that you can identify as an intact analyte in the mass spectrometer. So nice publication. You can look into that, and lot of information. Time won't allow me to discuss further on this one, but you can look into that. How a first dimension and second dimension looks like in the UV based approach. Followed that in the mass spectrometry. So traditionally, the hydrophobic interaction is another method people are using for the based on hydrophobicity. It's based on the stationary phase on the column, and also, of course, what kind of buffers you're using. You are using a reverse order here. The high salt at the beginning and the low salt, right? So essentially, traditional buffers are not mass spec friendly, but of course nowadays ammonia containing buffers are using. People are using, and it can be used along with the mass spectrometry, as I mentioned in this publication. Okay, so this is the more data on the same thing, on the charge variants, how it looks like on the particular monoclonal antibody. Now, T is another technology. So essentially, it will be very useful for many things because it can be used standalone and also along with the UV as well as the fluorescent detectors. Also, it can be used with the mass spectrometry so that it can give you more information on many types, and particularly when you have a complex charge heterogeneity, right? And sometimes, like in even heavily glycosylated form, also a better separate in the capillary electrophoresis compared to the liquid chromatography. Okay, now this is giving an example saying that so how a two this is a CG approach on the UV based approach method or detection, so that essentially you're looking at a like how you are looking uh, a mass both in mass spectrometry and non-mass spectrometry, two different molecules. One is innovator Ristova, and the other one is biosimilar. How it looks like. Okay, and of course the biosimilar looks like a lot more variant compared to the innovator. Now, if you combine the mass spectrometry along with the mass spec compatible buffer, this one is done non-friendly buffer. That's a ESCA buffer, and then if you combine with the mass spec friendly buffer, then you can get a nice mass spectrum of this molecule. Again, a application published on this one. You can look into that. There is a lot more publication, and this is a publication done by few years back, where it's showing that how a uh, like but there are many molecules are analyzed here, but like how a CIEF and ICIF like looks like, and this is just a good example of like say a CIEF and ICIF of uh, uh, basis band. Okay, so and then of course mass spec analysis of the same uh, different peaks in the mass spectrometry and simple time of flight mass spectrometry. So as I mentioned, there is many monoclonal antibodies are analyzed on this particular application in this publication and reported, so that how the charge variants are looking both in both UV, that's a CIAF, and as well as ICAF. So you can look into that for more details, how this analysis is done, and also like the CE mass spec is sometimes like much more sensitive than the LC mass spec. It can be like your nanospray mass spec and mass spec sensitivity can get. Now in the 2D LC also you can do the uh, online reduction, and you can get a sub-unit analysis. So there is all automated methods. You can just keep your wires in the autosampler and program in such a way that or fill the bottles with the your reducing buffer, and then it can automatically reduce and give the sub-unit information for your molecule. Now, with the interest of time, I may skip some of these slides over the time, but like go quickly, like maybe under 5 to 10 minutes time. So that essentially mass spec is very critical. I discussed some of this mass spec, but let me quickly go through a purely mass spec based approach. So this is a typical a reduced in a denaturing condition of the RPMS. Okay, so that you are looking into all the different glycoforms. In this case, niche monoclonal antibody, there's about 18 glycoforms can be identified in a single analysis, and this information everybody want to see, right? And with accurate mass. Now, comparing like how the native mass spec versus denaturing condition looks like. So like a denatured condition, like you can see very nice charge envelope, and but when you deconvolute, data looks more or less same. Of course, like there is a difference in sensitivity because uh, you may need a little bit more sample with the native condition, and also you can see the charge envelope is not as nice as denaturing condition, and it also shift into the higher mass, and particularly if you want to do native mass spec, you have to have a mass spectrometry that can go like depends on your molecule, depends on your molecule or molecular weight of the molecule, can go to like 10 to 12 thousand M/C. So a typical example, as I mentioned, sample preparation, separation, mass spectrometry, analysis. So let's look into a quickly like how a different types of analysis looks like. In this case, like either the IDS fragmentation that get the Fab and Fc region and light and heavy chains by reduction by using reducing agent, of course native mass spec, and comparing like innovator versus two biosimilars and how it looks like, and the softwares are there to calculate nowadays, like see compare the biosimilars versus innovator. This called mirror plots, and also like software automatically gives you the ratios and the relative quant of these various glycoforms, and like this one it gives the reduced subunit analysis, the same way light and heavy chain, and you can get the FC and Fab regions. Now, if you look into that trypsin digestion for that peptide analysis, traditionally it will be done with the your HPLC reverse phase HPLC methods, and it gives a peaks. And one has to really like go and look into that what it peak means, right? If you have a standard and you know that if you find a new peak, then you have to scratch your head on what is that new peak is, right? So particularly that can happen with the comparing with the innovator versus biosimilar or different batches of the biosimilars. Now, if you have a mass spectrometry, and actually you can get the similar peaks, but also mass information. And if you have a MSMS capability, then you can do sequencing. So in this particular case, as you can see here in the mirror plot, so that there is a innovator versus uh, uh, the biosimilar, and you can see there is a like extra peak, and one peak is missing in the innovator compared to the biosimilar. Now like you can see that there's a lysine, one extra lysine in the biosimilar compared to the innovator. So this is essentially based on the particular sequencing of this particular peptide and particular peak. Now, you can also compare in the mass spectrometry, get information identification. Follow that, you can also get the comparing different samples and also get the its relative quantitation. Right. In this case, like you can see that in the biosimilar 1 and 2 and innovator, said how a same peptide looks like in the 3 samples. So, like if you look into that, the peak 1 and the peak 5 4 and 5, that looks much difference compared to the peak 2 and 3. They are very similar. Now, you can, as I mentioned, you can get the the PTMs easily can be identified in the mass spectrometry. This is case oxidized versus unmodified, not that much of difference. Of course, there is a huge difference in the different samples, innovator and biosimilar. And this is a different peptide. In this case, it's like a bigger peptide. And then of course, like this is like showing that different types of PTMs, comparison, like how it looks like. These are all software driven, and no one has to do manually in this case. Okay, including the extra lysine here. Now, glycosylation important, as I mentioned, and different ways it can be done. And of course, we have the O and N glycan, and of course, it's abbreviated as everybody knows, G1F, G0F, and G2F, and all those things, right? There are different types of uh, glycans attached into the amino acids, based on amino acids called O and N glycan. Now, N-glycans are, easily can be released by the enzymes, and then you can do the analysis of these glycans, then what kind of glycans you have. And O-glycans are a little bit complicated to release, and then probably it may take some more time to even get kits. For N-glycan, one can easily release them and label them, and actually analyze them by using fluorescence or the mass spectrometry. Now they have, as I mentioned, they have the in its own effects. That you can see that they may have an effect on the ADCC or CDC functions of this monoclonal antibody based on the whether it's having a extra fucose or galactose in this molecule. Okay, so these are all showing that how it will be analyzed. So intact one is by the mass spec essentially, or the people can glycoforms cannot be separated by the simple HPLC. Glycopeptides you can actually separate on the HPLC, but also mass spec is an ideal it's ideal. But release ones you can derivatize using the either fluorescence or the mass spec case of tablet process. One can do the monosaccharides and also sialic acid. Now, kit-based approaches are very convenient, and I think everybody, if you are working in this area, everybody using the one or the other way kits rather than getting the individual reagents and labeling them and analyzing, because kits are much more convenient and faster. And in this case, like nowadays, you can do it for in a 1 hour for the entire relevant release and labeling of this glycans. Now, as it's shown at 1 hour kits, instant it's called instant PC, and you can actually and of course sialic acid for the sialic acid analysis. So you can do that in the either HPLC or the mass spectrometry approach. So here you are seeing that some comparison based fluorescence as well as mass spec data, and mass spec is really ideal because you can, based on the isotopic fidelity, one can actually get the formula and to detect what kind of glycan it is, the structure wise, right? One can get that. Now, as you can see in here, this is the mass spec data. Nowadays, database can be easily can identify in the softwares. One can go and process and identify what kind of glycans you have. Okay, here you are seeing two different images. One is your fluorescence detector. The other one is the mass spec based detector. So more or less, it looks same. Why more or less? It looks same, right? And even the quantitation looks same. So essentially, bottom line is mass spec one can identify and without database, I mean, the reference standards and with the HPLC fluorescence detectors, probably one need a standards, internal standards. Now, when you go to the intact analysis, like you may not be able to identify all the glycoforms, right? Glycan variants. Okay, because of the intact and subunit level, it has its own benefits and advantages and disadvantages. So now you'll be seeing that you're seeing more with the subunit level compared to the or fragment level compared to the intact and or the reduced. Now, if you go for the released glycan, you can see actually much better results, right? So sensitivity wise increases because now we are only measuring the glycans, released glycans. Now, one can also use the mass spectrometry for the glycopeptide analysis. Now, glycopeptide analysis also gives you additional information saying that where exactly these glycans are attached into the which amino acid is attached. Particularly if you have, for example, like this Bohr ends are there, uh, then it will give you an idea saying that which N is associated with the glycans based on the fragment ions. So in this case, like you can see that this particular N is N amino acid is involved in the modification, right? So if you take traditional CID or HCG based fragmentation, your sequence coverage is very minimum, right? You can get only partial sequence, and that's the reason people are combining with the CID, HCG, and yeah, ECG or ETD fragmentation, where ECG fragmentation one can get a better fragmentation and better identification. So and in this case, like actually, in fact, like two different same molecule, but same peptide, two different types of glycans, G0F and G1F. So like identified in different analysis. Now, as I mentioned earlier, if you recollect, so HPLC based methods can be switched into the single quad mass spec because we are adding a mass detector to the chromatographer, right? So simple adding a single quad mass spec along with your HPLC, along with the UV, one can easily like get a, of course, your single quad mass spec should have a mass range up to 3000 or something like that. So you can get the all kinds of your monoclonal antibody analysis can be done, and essentially, since mass spec is essentially called multi-attribute methods, so that one can reduce the number of HPLCs, combination of UHPLC and the single quad mass spec. There is an advantage because nowadays mass specs are really good at, actually, if you look into that, this particular application published very recently, your mass accuracy is really, really good with the single core mass spec. It looks like your high resolution mass spec, right? So it's like really ideal for any labs where they really want to identify the glycoforms in a routine base. Overall, like many instruments are required for the biopharmaceutical biomolecular characterization, uh, all the structures as a function analysis. And as you can see here, there are many instruments. Even in Agilent, we have like an FTIR, fluorescent detectors, and function analysis like cytotoxicity, flow cytometers. There are different types of instruments along with the HPLC and other things, right? So, so that if there's any time left, I just briefly want to touch with one more thing. But if there's no time, I can skip that right now.
Sure, Doctor. Sure, Dr. Vadi. We
But
can
okay,
spend some more
well,
time. Yeah, I think that's
yeah.
fine.
Okay, so I will just okay. Let me see whether I have that slide. Still, I have that slide. Let's see. Oh, I don't have. Okay, I think I don't have that slides. So it's okay, no problem. So I just wanted to mention here like what I was wanted to show is uh, the pad tools, like some of these analysis nowadays we can do in the both in R&D as well as in the in your manufacturing settings, called a real-time analysis both in upstream and downstream. So essentially, in upstream, like from reactor, you can take the samples, the sampling devices, send the samples to your HPLC. HPLC based analysis definitely can do that as a called what we generally call as a pad tool on an LC pad tool. It's called a direct analysis of the sample. And if you're doing a downstream, the samples from the various stages of your polishing stage, samples in a different fractionation level directly sent to the HPLC, and along with the mass spec or single core mass spec, and it can do a direct analysis called the on-NLC pack tool. And actually, in fact, nowadays it's getting more and more popular. At least I can say people are started discussion how it can be used, implemented. The reason is very simple here for the real time or a pack tool is it is going to significantly reduce your burden collecting the fractions and storing them and sending to the quality control labs, get the analysis done, bringing that back and making a decision what to do. So all this, sample analysis done in the real time or near to real time, and by the, and also like, in the reactor side, we can actually, on a real time, we can fix the problem. If anything, something you want to supplement with the culture media, you can actually do that, right? In the downstream, you can make a quicker decision to fractionation or pulling the fractions and go for the next step. So, and also a better quality data and also data integration, digitalization. So all this can be done. And I actually had a few slides, but I deleted that later because of the time. So this is what I want to convey the message so that there's something people are talking adoption of the pharma, biopharma 4.0. That's very important. Yeah. So I think if I can take any questions, if anybody has any questions, we can ask. I'll try my best to answer the questions.
Sure. So thank you, Dr. Vadi. So what we'll do is we'll now open the floor for some questions. If the participants have any questions, you all can drop it in the chat box, or either you all can unmute and have a direct conversation. There is one query from Webhub in the chat box, and do you want me to read it, Dr. Web? What?
If Vaibhav is there,
Yeah.
he can directly ask the question.
Yes, yes.
Vaibhav,
Webhub,
are
are
you
you
there?
on the call? Yeah.
Yeah, he's there. You can unmute and
Yes, I have requested to unmute.
okay.
Hi,
Yeah, Vaibhav.
can you hear me?
Yes.
Yeah.
Yes, you're audible.
Hi, Vadi, how are you?
Good, Vaibhav. Thank you. How are you?
Hi, Edna, good to see you also.
Yeah, thank you.
Vadi, I think we have received a capillary electrophoresis unit here in CBA recently. So for all of us, I asked this question because I think many of us would be trying. So just wanted to check what kind of expertise is required and how difficult it is going to be to develop a method on a capillary electrophoresis system, and if any specific skill set is required so that
No,
the person can
in
reliably establish that.
Yeah, WIPO, capillary electrophoresis is another type of separation technique, right? So one need to understand that compared to your chromatography, of course, like it depends on the instruments. You may get a kits, and sometimes like easy to use kit-based approach, and that can be easily follow the SOPs, and you can run the samples. And in general, generally speaking, it may require not not like mass spectrometry skills, but require some additional skills compared to the chromatography. But once it is there, once people can understand what is capillary process, and because it is done in like really a small scale, right? So it's like really tiny capillaries one can use for separation, and essentially like it can be easily learned, and not a major problem. I personally not, even though like I had opportunity, I don't want to use mass spec along with the mass spec when I was like sitting in the lab. But one can easily. I have seen that people are using a day in day out, so one can do that. Yeah, of course some readings are required, some training is required. With the minimum training, one can do it. Yeah.
Thanks, Vadi.
Yeah.
Thank
Yeah.
you.
Yeah, thank you. Any other queries that the participants may have? Yeah, we can spend a few minutes because Dr. Vadi has extensive experience in this field. He has worked immensely on the bench, and when I was when I had certain biopharma queries regarding to mass spec, I think he has been instrumental in clearing that out. So,
Yeah. I have a lot of experience in mass spec. Yeah, so I worked for many years on the mass spec. Yeah.
right. So yeah. Please feel free to drop your queries in the chat box. You can also unmute. Alternately, you can also reach out to us separately, and we'll be happy to answer any specific queries.
Correct.
So I think with that, we'll come to the end of the session. So Dr. Wadi, thank you so much. I think the topic was really vast.
Yeah, I know, I
And
know. It's 1 hour
in
not
the
possible. Yeah, yeah, yeah,
exactly
yeah,
in
yeah,
the
yeah,
limited
yeah,
time that we
yeah,
have,
yeah,
you
yeah,
you managed to give us a very
yeah,
nice flavor of
yeah,
all the
yeah,
tools. So
yeah.
thank you very much for taking the time out. I know you've been traveling, you've been busy, but I'm sure the participants have got a very good introduction to all of these techniques, and we'll only build on this in the future. To all the participants, I have shared our website details, our contact details with you all. Please do feel free to reach out to us for any of your specific biopharma characterization queries. I have also shared the feedback for this, the feedback link for this session. Please do give us your feedback because it will only help us on improvising the upcoming talks. So please do that. With this, I'd like to mention that the session has been recorded and we will be uploading it on our YouTube channel. I'll share the link details. I'll circulate the link details with all of you all via email, so you all can refer to it in the future. Dr. Wadi, would you be comfortable sharing the slide deck with us so I
Yeah,
can share it with the
yeah, yeah,
participants?
yeah, absolutely. Yeah, yeah. No
Sure,
problem. I'll share that.
sure.
Yeah.
Sure. So with that, we'll come to the end and we'll close this session. Thanks to the
Thank,
speaker, Dr. Wadi, and thanks to all of you all for also, being part of this session. And we'll meet you all next month with a new topic, and stay tuned for the, for that information. Thank you
thank you.
and have a pleasant
Thank you.
evening.
Bye.
Yeah. Yeah.
Bye.
Thank
Thank
you.
you,
Yeah.
Dr.
Bye.
Wadi.
Bye.
Bye-bye.
Thanks. Bye.